I'm Dr. Williams. I'm just going to go through some Biology Paper 1 with you. We're going to start unit by unit. This is the first unit, B1 cells. I'm just going to give you some tips and some advice and the content that you need to know before you go into your exam. If you're just starting on your learning journey with this unit, you might want to pause the video and make a few notes. But if you're the night before your exam and you're just doing it for quick revision, then just watch it in one go as a bit of a refresher. Right, let's get started. So for paper 1, you need to know about cells, and cells are the basic building blocks of all living things. So all living organisms are made of cells. In multicellular organisms, those cells are organised into tissues, and the definition of a tissue is a group of cells with similar structure working together to carry out a function. And then different types of tissues can work together to carry out a function as an organ. So for example, your heart is an organ made of muscle tissue and fat tissue, all working together to pump blood around the body. The organs can then be organised into organ systems, and that forms your whole organism. So the heart as an organ is part of the circulatory system. So all multicellular organisms are organised in this way. Plants or animals all have cells, tissues, organs, and organ systems. In terms of the cells as the basic building blocks, all cells, no matter what they're from, whether it's multicellular or unicellular organisms, all have cytoplasm, cell membrane, genetic material, and ribosomes. They can be grouped as eukaryotic or prokaryotic cells, but they all have those key parts. The difference between a eukaryotic and prokaryotic cell is that eukaryotic cells have a nucleus which stores the genetic material, whereas prokaryotic cells do not have a nucleus. I always remind my students to say pro means no, because prokaryotic is no nucleus. So let's look at the basic structure of an animal cell, which is a eukaryotic cell. You need to know these key parts. You need to be able to label them and also what they do. The words in bold are really important on this slide. If you look at the nucleus, the words in bold say controls the cell. So if you try to think of the word nucleus and associate it with the word control, then hopefully you might be able to remember what it does in your exam. The cytoplasm is where reactions happen. Mitochondria, that's aerobic respiration. Ribosomes, protein synthesis, and the role of the cell membrane is to let substances in and out of the cell. So they're all the core parts of a typical animal cell. If we look at a typical plant cell, they're also eukaryotic because they have a nucleus, but they have different parts to an animal cell. And those different parts are the cell wall, which is made of cellulose, and that supports the cell. It doesn't protect the cell, it supports it. It helps the cell, the plant, to grow into large organisms that they can be. They also have permanent vacuoles. They contain the cell sap, and they have chloroplasts, and their role is to carry out photosynthesis. If you look at another type of eukaryotic cell, yeast cells, again, you get those similar core features of the nucleus, cell membrane, ribosomes, mitochondria, and cytoplasm, just like plant and animal cells, but you also have a cell wall on the outside, which supports the cell again, but unlike plant cells, is not made of cellulose. It's made of a different material. You don't need to know that material, you just need to know it has a cell wall, and that is slightly different to the ones of plants. In terms of prokaryotic cells, our bacterial cells are the prokaryotes. You need to know that their genetic material is not inside a nucleus. So pro means no nucleus. It also means no mitochondria and no chloroplasts. It doesn't have those larger subcellular structures because it's about the same size as a mitochondria. So it can't have mitochondria within it because they are similar sizes. But it does have other key features that are unique to prokaryotes, which are plasmids. And plasmids are small circuits of DNA which contain a few genes, different to the genetic material because they are such a few number of genes on them. Okay, so they can actually be transferred from bacteria to bacteria, whereas the genetic material stays within that particular bacterial cell. They also have cell walls, again, not made of cellulose. And then some bacteria can all have a flagella, and that helps them to move. Flagella is not unique to prokaryotic cells, though, because eukaryotic cells like sperm cells, can also have a flagella. So it isn't a unique feature of a prokaryote, it's just something that some prokaryotes can have. You need to be able to compare the eukaryotic and prokaryotic cells. So this table is a really nice summary of that information. I suggest you pause it and have a look and check that you understand this information. And the key thing about eukaryotes and prokaryotes is their size difference. The eukaryotes are much larger in size, prokaryotes are much smaller. Prokaryotes have been around on Earth a lot longer than eukaryotes as well, so you could say they were older, but you can see that they actually do have key similarities and differences. It could be that you're asking long 6-mark questions to compare them. And if you're asked to compare, make sure you say that one has this, whereas the other one doesn't, to make sure that comparison is clear. All cells can become specialized, though. They were the typical plant, animal, yeast, and bacterial cells that you might see, but they weren't specialized to carry out a particular function. So when a cell becomes specialized, it has a particular role or function to carry out inside that organism. These are some specialized cells that you should know about and how they're adapted to carry out that function. You will come across them a lot throughout the biology course. I'm just going to quickly read through the cells, but you will learn more about them as you study further along in this unit. So sperm cell, nerve cell, muscle cell, root hair cells, xylem and phloem. They're all really important specialized cells that come up a lot across the biology course. So in order for a cell to become specialized, a cell must differentiate. As it differentiates, it acquires different subcellular structures to enable it to carry out a certain function. Now, you have to know that definition of how a cell becomes specialized. So the bits in bold, the different subcellular structures, and the certain function, they're the bits to remember in any description of how a cell differentiates. Most types of animal cell can differentiate at an early stage of their development. So that is during the embryonic stage, really, but some can differentiate later in life. Many types of plant cell can differentiate throughout their life, though. They don't have the same restrictions that animal cells have. We can see cells using a microscope, and there's a diagram of a microscope there with all the key parts labelled. The most important things to remember about a microscope, which enable you to use it, are the eyepiece lens. That's where you look down. That contains a lens with a magnification. The objective lens, that's also got a lensing with a certain magnification, and they can be changed and rotated. You've got the stage where you put the specimen, you've got your light, which passes through the specimen, and you have your focusing wheel, so you can clearly see the images that you're looking at. And microscopes allow us to see and understand subcellular structures. And there are two main types. The light microscopes that we use in the laboratories, they can view living cells, so you can look at key processes happening, but they have a low magnification and resolution. That means that you see things at high resolution, so it looks a little bit blurry if you're trying to look at smaller things like mitochondria. And they don't have a large magnification. Only up to about 2,000 times bigger can we see any cells that we put under a light microscope. Electron microscopes, however, are really useful at seeing those subcellular structures like mitochondria because they got a high magnification, about 1 million times magnification, and a high resolution, so you can distinguish between two separate parts quite easily. But the drawback is you can't view living samples, and they are very expensive to buy and use. There is a required practical to do with microscopy. From one of the years, you were given those pieces of equipment and you were asked to use those bits of equipment to measure the size of a cell. So a typical method for that would be to place the ruler on the stage, look down the eyepiece lens and focus on the ruler. You then measure the diameter of the field of view. That's what you can see when you look down the microscope. You measure that field of view in millimeters, and then you swap your ruler with the slide with your cells on, and you count the number of cells that you can see in that field of view, and then you can calculate a mean by dividing that millimeters that you've measured by the number of cells that you can see. And therefore, you've got the mean length of a cell. However, for some descriptions, you might just be asked to how do you view something under a microscope? And in that case, you have to talk about placing the slide on the stage, turning on the light so the light passes through the specimen and through into the eyepiece lens so you can see it. You use the lowest power objective lens initially to find the bit that you're looking for, and that's usually a ×4 magnification lens. And you look through the eyepiece lens and focus on your specimen, and then you turn the objective lens to a higher magnification depending on which magnification you want to use to look at your cells. As a top tip, if you're trying to remember how to view cells under the microscope and in that description, I suggest you think of this phrase, find on 4 and focus, which basically means you find your cells on the 4 times magnification and then you focus on your cells. That's your starting point for any description about using a microscope. If you're then asked to draw an image of the cells that you might see, you have to be able to do it in a scientific way. So the first thing to do is to include the magnification that you're drawing them at. So what is the magnification that the question tells you? And then you would draw a continuous line for any cell membranes or walls that you might see. If you can't tell the difference on the image, like the one shown on this slide, then you wouldn't draw that any different either. You would just draw it as one single line. You would only draw the key structures. So in the photograph, I can see sort of like little dark little spots inside the cell, but I wouldn't draw those because they're not a structure I can work out what they are. So I would just draw the nucleus. You wouldn't do any shading or kind of any roughness of your lines. You try and draw your cell a similar size to the original image, and you only draw the cell that the question asks you to draw. So don't draw all of the pictures that you can see. If it just says to draw one cell, just draw one cell. You will also be asked to use the magnification equation. Now, this equation is not given to you on the paper normally. It might be in a foundation paper, but it won't be on a higher paper. So you'll have to go into your exam knowing this equation. And the equation is magnification is the size of the image divided by the size of the real object. So sometimes you would get the word actual in place of real object, but that is the actual cell that you're looking at. The size of the image is the picture. The image is the picture or the diagram that you're shown. So the image on the screen shown there is 12 millimeters in length when I've measured it with my ruler. So that would be the size of the image, and the magnification could be given to you, or the size of the real object should be given to you, and then you would have to work out the missing value. Now, you can remember that equation as m equals I divided by A, but I like to just remember it as I am. So I equals A times n. It's a rearrangement of that equation, but it doesn't really matter how you remember it as long as you remember it in the right way. If you're going to get a picture on magnification, you would have to measure the images in millimeters. That makes it much easier to convert into the same units. So if you measure in millimeters, you need to convert to micrometers if you're given an image size in micrometers, or the other way around. So just make sure you're in the same units and use E now. That technique we use in physics applies here. Write the equation out first, insert your numbers, then write the answer to the question and give the units. Remember, there isn't really a unit for magnification. It's just an X normally. So don't worry too much about remembering that unit. Normally it's given to you on exam questions, but just draw an X if you're not sure and you're asked for magnification. In terms of conversions between millimeters and micrometers, you need to be able to convert easily between all of these units. And it's important to really remember it's by a factor of 1,000, depending on the direction or which way round you want to convert these units in. You've also got standard form there, so you need to be able to write your answers in standard form. So if you're not sure about that, I suggest you go look into that further, but that should be something you're familiar with from your other sciences and from maths. Now let's have a little look inside the nucleus of a cell for eukaryotic organisms. So we are going to look at the genetic material and think about how cells divide. So the nucleus of a cell contains structures called chromosomes, and they are made of DNA molecules. Each of those chromosomes made of DNA contains a large number of genes, thousands of genes per chromosome normally. Each gene, which is a small section of that DNA, codes for a protein, and that protein determines our characteristic. So, for example, you could have a gene on a chromosome, and that gene controls the cell to make a protein that determines the colour of your eye. In body cells, there are chromosomes in pairs, and in humans, there are 23 pairs of chromosomes. So the reason we need to know about this organisation is because when a cell needs to divide, it needs to replicate and copy those chromosomes in order to make new cells. So eukaryotic cells, which have a nucleus, divide in something called the cell cycle, and there are 3 stages to the cell cycle. The cell cycle is really important if you're to grow or repair your cells, or how multicellular organisms are going to develop and carry out their functions. So the cell cycle, the most important thing it needs to do is to double the genetic material and divide the nucleus. And there are 3 stages to it, as I said before: interphase, mitosis, and cell division, which we can also call cytokinesis. In terms of how the cell cycle happens and how a cell divides, you need to be able to describe this really specifically using some very key terms. I've tried to show them in bold on here, and I've also shown which stage they are in. So the cell shows, in this image, shows that there are 2 pairs of chromosomes. I've obviously not drawn 23, that would be very confusing. So we're just going to say that that cell has 2 pairs of chromosomes. The DNA, first of all, is replicated when the cell is ready to increase in size. And so if we're having more cells made from this process, we also need to increase the number of subcellular structures, things like mitochondria. If it's a plant cell, chloroplast, we need more ribosomes. So we are going to make more of those. We're going to increase the number of those structures. So the cell will also increase in size. That happens during the end of interphase. Normally, a cell in interphase is going about its daily functions. When it gets to the end of the interphase stage, that's when these things start to happen. The next stage of a cell dividing is that the chromosomes line up in the middle of the cell. And we know that now at this stage, mitosis has started. So as soon as we get mitosis, the chromosomes start to appear and move about in the cell. When the chromosomes are lined up in the middle, one set of each chromosome has been pulled to each end or pole of the cell. And we say the nucleus at this point has now divided. This is towards the end of mitosis now. So the next stage is cell division, where the cell membrane and the cytoplasm divide. And what you end up with are 2 genetically identical daughter cells. They're now in interphase. They are going about their daily functions. They are genetically identical to each other and the original cell that they came from. And those cells can then go on to divide themselves and go through this cycle again. There is actually a cell cycle dance that I do when I teach my students. That will be on a different video that you can go and watch, just to help you remember these key stages and the terminology that we need to use. So another feature of cells is their ability to divide in such a way. And if you think about the uniqueness of multicellular organisms, they come from one single fertilized cell and they manage to make billions of cells in order to make an organism. And the way they do that is that original fertilized cell is actually a stem cell. And as that cell develops into an embryo, the embryo has many stem cells in it. And a stem cell is just an undifferentiated cell that can divide itself and give rise to many more cells of the same type, or to differentiate into other different types of cells. So that basically means stem cells can divide to give more stem cells, or it can divide to make specialized cells. So they are the basic building block and the starting point of where all of the cells come from. There are 2 main types of stem cell: embryonic stem cells and adult stem cells. Embryonic stem cells, as the name suggests, comes from embryos, and they can differentiate or specialize into any different type of cell. So an embryonic stem cell, for example, could turn into a nerve cell or a blood cell, or a liver cell, or any cell that your body needs to have. Adult stem cells are found in specific tissues like bone marrow or the skin, and can only differentiate into certain types of cells. For example, the bone marrow stem cells can only be used to make blood cells. So stem cells can be really useful in medicine. They can be used in therapeutic cloning. So the idea is that you would take embryos with the same genes as the patient. You can isolate them and grow them into tissues that can actually be transplanted into the patient. This is really good because they have the same genes as the patient they're being transplanted to, so that they will not be rejected by that patient's body. You would expect rejection if you had a transplant of a tissue or an organ from a donor that isn't genetically identical to you. They can also be used in reproductive cloning. So if you want to make completely new organisms, that's just called cloning generally, but it's actually reproductive cloning, and you can start at the starting point of the embryonic stem cells. A quick process for that, it says quick, but actually would take quite a long time, is you would take your egg cell, you would remove the nucleus from the egg cell and insert the nucleus from the thing you wanted to clone into the egg cell, grow that into an embryo, and then you would have eventually an identical clone or copy. And there's obviously issues around using stem cells, especially human stem cells, ethical and religious issues with using embryos. The embryos often come from failed IVF treatments or people who no longer need those embryos because they've successfully had a child. So there's ethical rights and wrongs of whether embryos should be used for this purpose because it could develop into a human life. There's also problems with using stem cells because you can increase the risk of transferring a viral infection. Plants, on the other hand, a lot less ethical issues surrounding this because they have tips of shoots and roots where they have groups of cells called meristems. And at these meristems, that's where you would find those stem cells. So it says that the tips of roots and shoots have cells in them that can differentiate throughout the life of a plant. I could take the root meristem cells and actually turn that into a flower if I wanted to, because the ability to change into any type of cell is there with these plant meristems. You can use this really well if you want to clone plants quickly. You can save rare species. You can clone plants which have certain types of features that you want. So it's obviously a very useful technique in protecting species from extinction and growing crop plants. With the cells, we don't need to look at just their structures and how they grow and how they develop. We also need to look at how molecules move in and out of them. Now, this is quite a big part of any exam paper on biology. So I would recommend you really focus on knowing these three processes. So there are three processes of moving substances in and out of cells. They are diffusion, osmosis, and active transport. Osmosis is unique to water. So whenever a question asks you about water moving in or out of cells, you're thinking osmosis. Diffusion depends on your concentration gradient. So diffusion is the movement of molecules like glucose or urea, oxygen, carbon dioxide, amino acids, and things like that. However, if those concentration gradients aren't from a high to low, and you would have to move them against their concentration gradient. So from a low to a high concentration, you need active transport. So those same molecules can be moved, but it depends on how concentrated they are in or out of the cell. So let's have a look at each one of these in turn. We're starting to think about the movement of substances through specialized exchange surfaces, and these molecules will move along these exchange surfaces in plants and animals. And there's certain features of specialized exchange surfaces. So by this, I mean things like in the lungs, the alveoli, in the villi, in the small intestine, or leaves of plants. So all of these are exchange surfaces and need transport of molecules by diffusion or active transport or osmosis. So they have these features of a large surface area. They have features of a thin membrane that is usually 1 cell thick. That provides a very short diffusion path or a very short pathway for these molecules to move through. In animals, you'd want a really good blood supply that helps to maintain a concentration gradient. And you'd also want well ventilated in animals because that maintains concentration gradients if you're moving gases. Diffusion is actually the movement of particles from an area of high concentration to an area of low concentration. And that's the movement of particles of oxygen and carbon dioxide, glucose and urea. Diffusion is not unique to cells. Diffusion is something that can happen in chemistry as well, and it basically relies on that principle. So as you can see, there are molecules at very high concentration on the left-hand side of the screen, and on the right-hand side of the screen, much lower. So the molecules will move by random motion across to the right-hand side. And that is diffusion. If it happens across a cell membrane, then that's usually with substances like oxygen and carbon dioxide and glucose and amino acids. And different things can affect the rate of that diffusion. We talked about specialized exchange surfaces, but also things like concentration gradient. A bigger difference in concentration gradient, the faster the transport will happen. Temperature, higher temperature means faster transport because the particles have more kinetic energy to move across. But temperature isn't something easily changed inside an organism, so just bear that in mind. Surface area of membranes, the larger the surface area, the faster the transport. That's why we have specialized exchange surfaces. And the thickness of the transport path. So you can't make a cell membrane any thicker or thinner than it is. All you can do is change the number of cells that something like oxygen would need to pass through. So that's why a lot of specialized exchange surfaces are 1 cell thick to make that diffusion path as short as it can be. So you'd only have 1 cell to pass through. And the fewer the cells, the faster the transport. So diffusion all of those things are really important to maximize rates of diffusion and also active transport when we look at that, and osmosis. Another thing to bear in mind when you're looking at transporting of molecules, mainly by diffusion, but osmosis and active transport as well, is the surface area to volume ratio. If you're a small organism like that worm, then you can probably have a large surface area to compare to your volume and absorb oxygen through your skin and get rid of carbon dioxide also through your skin. So that allows all of the cells to get the oxygen that they need. However, as you increase in size, you need to have specialized exchange surfaces because your surface area to volume ratio gets smaller. So as you get bigger, that ratio gets smaller, you cannot get enough oxygen to diffuse through your skin, which is why we adapt specialized exchange surfaces. So that's important for organisms of certain sizes, and it determines how big you can grow without exchange surfaces. You might be asked to calculate surface area and volume, so you should be familiar with those calculations from maths, but just check you can do that before you go into the exam. Osmosis, then, if we think about that in particular, is the movement of water molecules through a partially permeable membrane from a dilute to concentrate solution. So you do need a membrane for osmosis to occur. A top tip to remember that definition, and you have to remember it like that, is Washington, D.C. Washington, water, D, dilute, C, concentrated. And you do need to remember it like that rather than thinking about concentration of water molecules, because what you can do is end up getting in a real pickle with the kind of words that you're using. So please learn the osmosis definition as the movement of water molecules through a partially permeable membrane from dilute to concentrated solution. A partially permeable membrane means it will only allow certain molecules to pass through it. And substances like sugars are often too large to pass through membranes, and it means that water can. If you look at those diagrams, just be familiar that a dilute solution means that there isn't much sugars or salts or any other solute dissolved in it. So a dilute solution wouldn't have many salts in there or sugars. So water will move from where there's a dilute solution to the more concentrated solution, which has more salts or sugars in there. And if we're thinking about our exam technique, this kind of description, whenever you get a question about osmosis, is a really useful one to learn. All you would have to say is, depending on the question, where is the water moving from and to? State that it's through the partially permeable membrane and talk about which solution is dilute and which solution is concentrated. So a useful phrase to learn is water moves, and you choose either into or out of the cell by osmosis through a partially permeable membrane from the dilute cytoplasm to the concentrated solution, for example. And there's an example there of the red blood cell. And if you had to explain why the cell bursts, you would say water has moved into the cell by osmosis through a partially permeable membrane from the dilute solution to the concentrated cytoplasm of the cell. And that would get you all the marks for a question like that. And then explaining why the cell shrinks, it's just the opposite. So water moves out of the cell by osmosis through a partially permeable membrane from the dilute cytoplasm to the concentrated solution. So it depends on the scenario, but you would be able to get all marks using that phrase. There is a required practical with osmosis, and I would make sure that you're familiar with the method for this. But most familiar with all of your required practicals is if there are independent, dependent, and control variables, that you are really clear about them. Now, it might be the required practical, if it is assessed in an exam, is not going to change the concentration of salt solutions as you did when you did this practical. It could, for example, change the temperature. Any of those control variables could be altered. What happens is if you change another factor, then all of those other things become the control variable. So in this example, the concentration of salt solution is your independent variable. That's what you change. You measure the change in mass, so the mass of the potato before and the mass of the potato after. Control variables like the cork bore they use to give the surface area, the volumes of the solutions that you put those potatoes in, the time you leave them to incubate, and the temperature at which you're incubating them are all important, and you must keep those the same. When you do this investigation, another key control is when you take your potato cylinders out of the solutions, you should roll them onto a piece of filter paper or paper towel in order to remove excess water, but don't leave them on the filter paper because it will absorb water out of the cells. So that's a really important control variable as well when you're measuring your masses of your potato before and after. And you can record your initial mass and your final mass, work out the change in mass, and then the percentage change. It's much better to work out percentage change because your starting masses might all be slightly different, even if you cut your potato to the same length. So measuring a percentage change in mass allows you to compare the different results much more easily and more with more validity to your data that you collect. When you've worked out the percentage change in mass, you will be asked to plot a graph. So when you're plotting a graph, there's certain rules that you need to follow, and that's true in chemistry and physics and everywhere. And when you plot a graph, you always put your independent variable, that's the one that you've changed in your investigation, on the x-axis. And this variable must be evenly spaced. You don't just although this is the numbers given on that graph that you've drawn, you don't just write the numbers given, you make sure they are evenly spaced when you draw your independent variable. And make sure you label it really clearly, use the titles that are given to you, and make sure you have units. That's usually worth a mark or 2 marks inside an exam paper, so make sure you're getting that done first of all, and that's accurately done as well. Excuse me. Then you must accurately plot the points in pencil, and you use an X. The X marks the spot. Don't do any circles. Use a pencil just in case you make a mistake. And plot all those points accurately. That will be checked by examiners, so make sure you are checking that carefully as you do them. And then you always have to draw a line of best fit. Now, a line can be any shape. It can be a curve. It can be a straight line. What you have to look for are any anomalous points, the ones that don't fit the pattern, and discount them. But in this example, there are no anomalies, and you would draw a curve going through your point. And it has to be smooth. Do it in pencil in case you make a mistake. Rub it out completely and do it again. Make sure that you are going through the points. You don't just, sidestep any, even by a slight bit. And if you make a mistake, it is okay to rub out and just do it all completely again. Try to do any curves in one smooth movement if you can. Now, this is really important to do accurately because you could use this graph to work out the concentration inside your potatoes. So different parts of the graph mean different things. At the top, it says the water has moved into the cells of the potato by osmosis through a partially permeable membrane from the dilute solution to the cytoplasm of the cell. So the potato gained mass. The cells don't burst in this example because plant cells have cell walls. So the mass is increased in my potato because it's in a dilute solution. At the point where you cross that x-axis, that shows there is no net movement of water into or out of the potato by osmosis. You have to say net movement because the water molecules will still move by random motion, but there is no overall movement in or out. The concentration of the cytoplasm at that point is the same as the concentration of the solution. So where it crosses that x-axis at 0.28 or 9, I think, you would say that that is the concentration of the cytoplasm of your potato cells. And you just read off the x-axis to know that. Below that line, the water's moved out of the cell by osmosis through that partially permeable membrane from the dilute cytoplasm to the concentrated solution, and the potato loses mass. So it's really important you understand those key features of that osmosis practical and how to interpret the results that you get. The very last way of transporting molecules in and out of cells is active transport. And this is basically the opposite to diffusion, because what it relies on is substances moving from a low concentration to a high concentration against the concentration gradient through a partially permeable membrane. And this movement of molecules requires energy released from aerobic respiration. So cells like those you'd find in the villus, those sorts of cells will have lots of mitochondria to release the energy needed for active transport. Or cells like root hair cells, which require active transport to move the nutrients from a low concentration in the soil to a high concentration in the cell, they would have many mitochondria because they would need the energy to be released for the active transport to take place. So that's often a clue. If a cell has a lot of mitochondria, it might mean that it needs a lot of energy to be released for active transport of molecules against their concentration gradient. Okay, that is it for paper 1B1 cells. What you'll need to do now is maybe do some Quizlets to recall that knowledge. You could do some practice questions, or you could just watch the video again. But whatever you choose to do next, good luck for your exams, and I hope this has helped.